Okay, it's 3 o'clock or 2 o'clock or 1 o'clock, depending where you are. So hi everyone, and again, let's let people come in as they are ready, but let's slowly start. So welcome to the second HypoxEU event. My name is Krista Rantanen, and I'm the Director of Scientific Applications here at Baker Ruskin, but today I'm your host. And today I'm in Finland. It's comfortable, minus, a few degrees centigrade, slow snowfall, and very Christmassy here indeed. However, I'm not alone. Joining me today, not from Finland, but from UK, are two people. And one of them is the surprise scientific moderator, Tammy Bishop from the University of Oxford. So Tammy will be the one that's going to take your questions and moderate them, and also keep an eye on the time. So our speakers have now been given 20 minutes, and then there's a Q&A. So that was requested after the first HypoxEU event. So now we want to have time for the questions as well. So Tammy here, thank you, is going to be doing that job. And then another surprise guest that we have today here is my friend, Professor Giovanni Mann from King's College London. And Giovanni is going to say a few words to open this HypoxEU 2. So Giovanni, please.
Thank you very much, Krista, and thank you for everyone who's registered for the meeting. It looks quite good. We have 138 participants, so that's exciting. The program looks good, and I speak from King's. My sin for joining you is that I'm interested in hypoxia and ischemia reperfusion injury. We have a very elegant set of Baker equipment here to do work with vascular endothelial and smooth muscle cells. And we've identified quite clearly that if cells are maintained at physiological normoxia, that's the normoxia that a cell or tissue experiences in vivo, that the redox phenotype of these cells is dramatically different. Another clue for those that are interested in HIF-1 alpha and other HIF factors is that if you have those cells adapted long-term, and I'm talking 5 days, at a given oxygen level that's physiological, you can not have HIF-1 alpha stabilization. So we can look at the real physiological phenotype. That's just the sales pitch for which we have. I welcome any of you to come and visit us at King's, but now I'd like to hand over to Krista and say, let's start an exciting scientific program.
Yeah, there we go. Thank you, Giovanni. Just a few housekeeping notes how this is going to go today. Again, I'm going to keep everybody muted. So that's one. This whole thing is being recorded, so I hope that's okay. And if you have questions, and when you have questions, please put them in the chat box so Tammy can pick them up from there. And then after each talk, we can go over few, maybe not all, but they will again be answered later. So to the actual program here. First, we have Carsten here, and I'm just going to again introduce the speakers a little bit more in depth. So Carsten is originally from Bremen, and for those of you who don't know, Bremen is a city in northern part of Germany, and also it's very famous for that town musicians of Bremen. And at least when I was growing up, that was the fairy tale that I remember quite well. So thank you, Carsten, for reminding me. I forgot that one, but that's true. That's from there. So that's where he's from, and he did his PhD and postdoc with Professor Cormac Taylor there somewhere in the audience at the University College of Dublin. And after this, he became a group leader with Professor Roland Wenger. Also here, I can see you there, also in our scientific organization committee, by the way, as is Cormac Taylor. And is now at the University of Zurich. So Carsten mainly studies how cellular oxygen sensors confer hypoxia adaptation to cells and tissues, and how this in turn could be utilized in the treatment of hypoxia associated diseases. He has 2 young kids, and I believe that keeps you quite busy, but then he says that he does these odd mountain bike trips in the nearby hills that are there around his university. But Carsten, I'm thinking if you're working with Roland there, and he seems to, and I remember being quite a huge fan of hiking, and so I can imagine your mountain biking isn't too bad either. So, okay, Carsten, the floor is yours, and I'm gonna go mute here, please.
Yeah, thank you very much, Krista, for this very nice introduction. And thank you to all the organizers for inviting me to present here today, which is a great opportunity at this brilliant seminar series, shown again with a huge number of participants. So thank you very much. And if you wonder about my background, so I'm currently actually in a hotel in Greifswald in northern Germany. So this is not in my office, this big picture, but it's in a hotel. And I'm talking now today about hypoxia sensing and signaling. And we all, of course, know the typical oxygen sensors, so the PhDs or prolyl hydroxylases of the 2-oxyglutarate-dependent dioxygenase superfamily, which hydroxylate the proteins in HIF and lead to its degradation. And we, of course, also know the asparagine hydroxylase FAH of a different subfamily, the Jumanji C subfamily, which also regulates HIF alpha. And now in the recent 2 years, there were also further discoveries of other cellular oxygen sensors, so the KDMs, which actually belong to the same subfamily as FAH, which regulate histone lysine demethylation, and therefore epigenetic regulation is affected in an oxygen-dependent manner. And we also learned about now other cellular oxygen sensors like ADO and PAM. So therefore now our knowledge is ever so increasing about the cellular oxygen sensors, and there's more than we knew for the past 20 years. And what I will now propose during my talk is that there are not only more oxygen sensors than we have known so far, but there's likely also other mechanisms of the known oxygen sensors which likely contribute to hypoxia sensing and signaling. So, and my group in the last few years has especially focused on FAH, and as said, FAH is of course best known for the regulation of HIF-1 alpha, but it has also been shown that it can regulate other proteins outside the HIF pathway. And we also know from work from Randy Johnson's group that the FAH knockout leads to hyperventilation, energy, metabolic phenotype. But what we don't know really is the enzymatic, the link between the enzymatic activity of FAH and this phenotype. So it may be in part through the regulation of HIF-1 alpha, but it is likely also through the regulation of other proteins. But we don't really understand now how this metabolic phenotype is coming to terms. And what we have previously shown is that FAH is hydroxylating the deubiquitinase O2B1, which leads to a differential regulation of cellular energy metabolism, which was of course for us highly intriguing. And therefore we also analyzed O2B1 knockout mice for their phenotype. And here we also observed that these mice have also hyperventilation and a very comparable energy metabolic phenotype to the FAH knockout mice. So this supports now our current hypothesis that the regulation of O2B1 by FAH is at least contributing to the phenotype here we observe in mice. And we then analyzed now also the interaction between FAH and O2B1 on the molecular level in detail. And what we found, much to our surprise, was the following, what you can see here in this immunoblot. So here are 3 samples ectopically expressing FAH, and in these 2 samples additionally expressing the deubiquitinase O2B1. And here as wild type, and here with a point mutation in the hydroxylation site. And you can see that here an additional band occurs at a high molecular weight when both FAH and O2B1 are expressed here. But this is disappearing when the point mutation on the hydroxylation site is present, and we can detect this high molecular weight complex both with an antibody against the FAH and against O2B1. So we could then show with mass spectrometry and other methods that this is indeed a complex of FAH and OTB1, which survives the denaturing conditions of SDS-PH. And we could then also purify it from bacteria, and we performed a range of different biochemical analyses. And no matter which chemical we use, we cannot destroy this interaction. So therefore, we propose that this is a covalent bond, which is formed between FAH and OTB1. We analyzed that in more detail and looked at it at different oxygen levels, as you can see here indicated. We also looked at HIF1 alpha and HIF2 alpha stabilization in the very same samples. And here the effect on this heterodimer and its formation. And if we then quantified it over several experiments, and we looked then at what oxygen levels do we have 50% present of the HIF1 or HIF2 alpha protein and of the heterodimer to get a sense for the hypoxia sensitivity. And this is shown here. So for this heterodimer, we had 50% loss, I should say, here at 4.1% oxygen. And this was highly surprising because so far it was known that the FAH-dependent of HIF1 alpha is only affected below 1%. So this means this heterodimer formation has a much higher hypoxia sensitivity than FAH-dependent hydroxylation of HIF1 alpha. And it appears to be even more sensitive than the proline hydroxylase-dependent regulation of the HIF alpha subunits. So therefore, this complex is formed via the asparagine 22 of OTB1. It depends on FAH activity. Also, when we genetically inhibit FAH activity, we prevent this formation. It's highly hypoxia sensitive, and we could show that it occurs co-translationally. It's likely covalent and affects also the OTB1 deubiquitinase activity. So, and this was very intriguing to us, and therefore we asked the question, so is this kind of complex formation specific for FAH and OTB1? And for that, we designed an experiment, but we took an experiment basically from the ubiquitin system where people, when they want to show that a ubiquitin is covalently linked to a substrate, they perform such an experiment. So here we now adapted it for our purposes. So basically, we performed an IP of the OTB1, and so here's just the control, and with FAH together ectopically expressed, and we then had a normal IP with native lysis buffer, and we could observe the complex and also the normal interaction here of FAH. And then here we used 1% STS and boiling to denature the lysate prior to the immunoprecipitation, and therefore disrupted all non-covalent interactions. As you can see here, we lose the normal interaction, but the covalent interaction is still present. So therefore, we now had a way of how we can distinguish between normal and the novel covalent type of interaction. So therefore, we performed this experiment again, but used mass spectrometry for the analysis. And this is shown here. So we have the native and the denatured lysates, and then we looked at proteins which were present under native conditions and which survived the denaturing conditions and therefore present in both samples. And you can see here we found 13 proteins, so indicating that FAH indeed forms such complexes also with other proteins. It was very reassuring that we found again OTB1 within these 13 proteins. So confirming our previous results that this is a covalent complex. So, and because now we saw that there is more than just one of such complexes within the cells, we decided that we name such complexes, and we call them now oxomers, or oxygen-dependent, stable, or likely covalent protein oligomers. So therefore, I will refer to these complexes now as oxomers. And when we looked at these putative other substrates for oxomer formation, one protein jumped out to us, which was this one, I-kappa B beta. So I-kappa B beta is a negative regulator of the pro-inflammatory transcription factor NF-kappa B. And this was interesting to us because we had previously shown that FAH can regulate the pro-inflammatory IL-1 beta signaling pathway and affects IL-1 beta dependent NF-kappa B activity. And we then, in that previous analysis, we analyzed or investigated how FAH may be able to do that. And there we found OTB1. This is how we originally came to identify OTB1, and how then subsequently we could show that it's indeed also hydroxylated. And in the very same screen, we had then also identified I-kappa-B beta as interaction partner of FIH. And there we also identified a putative hydroxylation site. So this was oxidized in our screen. We don't know whether it is hydroxylated, but we also had a putative oxidation site. And now this was a screen 7 or 8 years ago in a very different lab. I was a PhD student with Cormac Taylor. So all completely different questions and completely different people involved in the experiment. Now, 8 days later, we perform a screen for a very different reason. And we find again, I-kappa-B beta as partner of FIH. And therefore, together with the knowledge about the regulation of FIH of the pro-inflammatory signaling, we said we have to analyze this in more detail. So, and this is now what Julia Volkova is doing, a PhD student in my group. And she ectopically expressed FIH and I-kappa-B beta together in all of these samples at different treatments. So here in Normoxia, you can see, so here I-kappa-B beta is expressed, so here in all samples, and also here FIH in all samples. And then we observed here also, again, a higher molecular weight signal, which is detected with an antibody against I-kappa-B beta and against FIH, as shown here. And also when you add up the molecular weight of I-kappa-B beta and FIH, it also fits to the molecular weight we could detect here. So therefore, FIH and I-kappa-B beta indeed also form an oxomer, which is also shown then when we use hypoxia, we inhibit completely the formation of this complex. And when we then use, instead of hypoxia, DMOC, a pan-hydroxylase inhibitor, we can prevent the formation of the oxomer. And also with DFX, an iron chelator and a pan inhibitor, but when we use FG4592 or roxadustat, which is known as others, and also we have shown this is specific for the prolyl hydroxylases or PHDs, this does not prevent this oxomer formation. So therefore, clearly depends on FIH activity. We've also shown that when we mutate FIH, so that's not catalytically active anymore, that also prevents this formation. So I-kappa-B beta belongs to a family of proteins, the I-kappa-B protein family. So I-kappa-B beta is here, but was previously shown by the group of Peter Radcliffe that FIH interacts and hydroxylates other members of this family. So on the one hand, I-kappa-B alpha is hydroxylated on 2 asparagine residues by FIH, as is P105 as on 1 asparagine residue, and also I-kappa-B epsilon is an interaction partner for FIH. And has also been well established through the work of Peter Radcliffe, Chris Schofield, and others that FIH interacts with ankyrin repeat domains and often hydroxylates them. So now we identified I-kappa-B beta as also interaction partner and as an oxomer forming partner of FIH. And I-kappa-B alpha and I-kappa-B beta are the most similar proteins of this family. They're highly similar, as you can see here in this picture. And we were also interested in how similar are they to each other on a 3-dimensional structural level. So the problem was that there's no structure for human I-kappa-B beta available. So therefore, we initially could not perform such comparisons. But recently, there was this publication just a few months ago about the database AlphaFold, which is from EMBL and together with the artificial intelligence of Google, DeepMind, and they can actually predict to a degree which has never been possible before the 3-dimensional structure of proteins. So we decided we use that and now looked into the structure of I-kappa-B alpha and I-kappa-B beta. You can see here the ankyrin repeat domain, nicely shown here and here. And then we overlaid them together to see how similar are they to each other. You can see the ankyrin repeat domain are basically identical. If you also look here from the top between green and cyan, I find it very difficult to see any difference here whatsoever in between the ankyrin repeat domain. And when we then look at the actual hydroxylation site of I-kappa-B alpha, that's shown here in yellow. These are the asparagine residues that are hydroxylated in I-kappa-B alpha. They lie here. And then for comparison in red, we highlighted all asparagine residues of I-kappa-B beta so that we can compare them. And you can see, maybe not exactly in the same, but in a very similar area, there is also an asparagine from I-kappa-B beta. So it looks like this would also be accessible for FAH. So thus I kappa B alpha and beta are really similar to each other. Therefore, we asked the question, can I kappa B alpha also form an oximer with FIH? And for that, we then wanted to have them as comparable as possible. So to express I kappa B alpha or I kappa B beta from the very same type of vector with the same tag so that we can use the very same antibody to detect them. And that's what we did. And this is the experiment. So here you can see our control, and then here I kappa B beta overexpression and I kappa B alpha, which are very similar to each other, and then FIH expression here as well. So you can see I kappa B beta with FIH expression forms again this oximer as seen here, while I kappa B alpha, there's no sign whatsoever of any oximer formation despite this high similarity. So this shows that you cannot just express a substrate of FAH together with FAH and you will have an oximer formation, but it appears to be very specific to a subset of proteins, which we don't know yet and don't understand how or why is that, but it really seems to be very specific for a subset of proteins. And we next then also analyzed, so to which amino acid is now this oximer formed? And as we know from FAH and O2E1, it's to an asparagine. We therefore mutated each asparagine of I kappa B beta and to investigate whether this disrupts the oximer formation. And that's shown here. And we also, in addition, point mutated the aspartate we had originally identified as oxidized within the I kappa B beta protein. And so here you can see the expression of wild type I kappa B beta and the oximer formation. And none of the point mutation disrupted the oximer formation. So that was very surprising because this means that oximer formation between FAH and I kappa B beta does not occur through an asparagine. So FAH has been shown that it can also hydroxylate or modify other amino acids. So therefore, we of course now will expand our analysis towards aspartate, histidine, and others. It means it's not just between FAH and an asparagine residue, such an oximer formation. And there's actually also evidence in the literature for that this is also not just confined to FAH. Because this is now an investigation, again, from Peter Radcliffe's group and Matt Cockman. They analyzed OCFOD1, a member of the 2-oxyglutarate-dependent dioxygenase superfamily. And this hydroxylates RPS23, a small ribosomal protein, and exerts translational control through this hydroxylation. And this is a prolyl 3 hydroxylase, not a prolyl 4 hydroxylase like the PhDs, a prolyl 3 hydroxylase, but it's actually the most closely related human enzyme to PhD 1, 2, and 3. It's closer related to PhDs than FAH. As I showed you in the beginning, FAH belongs to a different subfamily of the superfamily. And in this investigation, they showed that here in an IP of OCFOD1 and an analysis via Kumassi-stained gel, that there was a higher molecular weight complex which actually consisted of OCFOD1 and RPS23, which survived the denaturing conditions of an SDS page. And also they could show that such a complex formation did not occur when they used DMOG or an iron chelator to inhibit OCFOD1 activity. So highly similar to what we've shown now for FAH and O2E1 and FAH and I kappa B beta. And that raised then for us the question, so can also the PhDs form such oximers? And for that, Agnieszka Jucht, another PhD student in my group, performed first a kind of look-see experiment where she overexpressed PhD 1, 2, or 3, or FAH, all with a V5 tag, so that we can use one antibody again, and either in the absence or presence of an iron chelator. And here you can see the overexpression of these proteins. And then we looked at higher molecular weight of forms, which were affected by DFX treatment, which inhibits the PhDs. So you can see here for PhD1, there were actually higher molecular weight bands, which were decreased or not formed in the presence of DFX. For PhD2, it's less obvious, but here was also something. For PhD3, it was again much more obvious. And for FAH, we knew that already from our previous analyses. So there were indications that also the PhDs can form oximers, and therefore we then performed the same spectrometry-based screen we had done with FAH for the PhDs, together with Alex von Kriegsheim in Edinburgh. And again, we used native and denatured lysates prior to the immunoprecipitation. And then we found several proteins which were present in native and denatured lysates. And we went here very strict then on to our definition, what is an oxomer, because we really said the protein should not really change in its abundance in native or denatured lysates. So if there was a too strong difference in fold change difference in between, we did not consider them as oxomers, even though they were present in both lysates. So therefore, we have here 22 oxomers identified, or likely putative oxomers for PhD1, 14 for PhD2, and 9 for PhD3. And what was reassuring, what we found with PhD3 was that among our putative oxomers, there was SUMO 234. And work from Eder and Berra had previously shown that PhD3 is SUMO-related, and this is a covalent modification. And we could now identify this covalent modification with our screen, again, confirming that we are here selecting covalent interactions. So, and now Agnes only started now to analyze this via immunoblotting, and this is a very preliminary data. Just to show you one example, so here we have the deubiquitinase, again, surprisingly, deubiquitinase USP15 and PhD1. And when we expressed both, we observed here a higher molecular weight signal, which we prevented when we used roxadustat, a PhD inhibitor. So therefore, it looks as if here PhD1 and USP15 form such an oxomer. So, and with that, I'd like to summarize. So we could show that FAH forms an oxomer with OTB1 and with I-kappa B beta. OCFOD1 and RPS23 are likely also forming such an oxomer, and as well as the PhD1, 2, and 3, where we are now analyzing the exact substrate proteins. And so this formation depends on hydroxylase activity, which was shown for all of these complexes. And it's highly hypoxia sensitive. This was really shown for FAH and OTB1. We are now currently in progress of analyzing it also for these other complexes. And for FAH and OTB1, we could show that such complex formation affects the enzymatic activity of OTB1. So therefore, because it also has an effect on the substrate protein, we propose now that oxomers are a new type of hypoxia sensing and signaling. Of course, we will now need to do more experiments in order to figure out in detail what is affected by this oxomer formation. And with that, I would like to thank all the people involved, so all my former and current group members, also Roli for all of his support for hosting us, and he's a great mentor who allowed me to do all of this work there in his lab, and all his also all the group of Professor Wenger, Roli, and all of our collaborators as well, and also Cormac for all of his mentorship throughout the years. And thank you very much for your attention. I'm happy to now answer any questions.
Thank you, Carsten. Yes, go ahead, Tammy. Yes.
Thank you, Carsten. So I have a first question in the chat from Daniel Dietrich, who's asking, what is the half-life of the oxomers, such as FIH and OTB1?
Yes, it's actually a rather surprisingly long half-life. So we looked at it, so the formation seems to take a while because it works through co-translation. So when we inhibit the translation, the formation doesn't occur, and therefore it takes some time to form, but it also is quite long. So between 24 and 48 hours was until we didn't see any more oxomers being present. So this is, we don't know exactly the half-life. I think it was between 10 and 16 hours roughly, but until we don't see any oxomers anymore, it takes up to 48 hours. So they're rather long-lived.
I don't know if anyone else has any questions. If you could.
I mean, I have a question, Peter.
Yeah.
You hear me? Very nice, Carsten. Now, one piece of information to put on the table, which might be relevant, and one question for you. The piece of information is that we've found that in some ankyrin repeat proteins, that there's more than one hydroxylation at the target site. The target site being, say, VNVN each asparagine is hydroxylated. So the question for you is this true of I kappa B beta, which I don't know. But the interesting thing in relation to what you've shown is that this double hydroxylation does not occur with a peptide substrate. It is completely specific to incellulo or the presentation of a polypeptide sequence, implying that there is a second binding site which is away from the catalytic site. So that allows us other possibilities that these crosslinks could be generated without the integrity of the asparagine hydroxylation site being intact. So that's a comment. The specific question is, what is the sequence at the target site?
So for I kappa B beta, so yeah, we haven't performed the mutation of several asparagines together within one I kappa B beta protein. So therefore, we don't know whether there are several hydroxylations occurring, and maybe that our single point mutation.
Is there more than one asparagine, Carsten?
On one asparagine, you mean more?
The unmodified species, does it contain more than one asparagine?
The I kappa B beta does contain 5 asparagines.
In close vicinity to the target site?
No, they're all scattered throughout the protein. There's no direct vicinity like in I kappa B alpha. So there we have these two close together structurally, but not in I kappa B beta. They are not close to each other at all. So it's actually, if I can go back, based on the structure. So it's these 5 are here, and here are these 4, and then there's another one here. So these would be probably the closest to each other, but there's none like here for I kappa B alpha, which are very close to each other in relative terms to the red and for I kappa B beta. And we don't know exactly where FAH is targeting I kappa B beta. We don't know that yet. So that's where we now have to analyze that further. Therefore, I can't say anything about the specific target site, unfortunately.
Thank you.
We have a question here from Sasha Benneker, who's asking whether all the substrates identified so far contain ankyrin repeats.
No, actually not. So for this oxomer formation, only I kappa B beta contains an ankyrin repeat.
Sorry, could you hear that?
So therefore, sorry, can you, therefore, it appears at the moment rather independent of ankyrin repeats if I kappa B alpha doesn't form and I kappa B beta does form it. But this is, of course, a rather vague hypothesis. We don't know for sure yet.
Tammy, can you hear us? Perhaps a bit bad connection there.
I can hear you. Can you hear me?
We can hear your answers, Constant. Yes, yes.
Okay, yes, yeah.
Yeah, okay. I can take this next question from Norma Masson to how can you investigate these oxomers in the native setting? Can you IP the endogenous enzymes from extracts portion based on size?
So we could show for FAH and OTB1 that it's formed endogenously, where we performed an immunoprecipitation of endogenous FAH and then looked for this complex formation. And actually in the work from Peter Reckless group, they showed that for OCFOD1 and RPS23, it's also occurring on the endogenous protein level. For the others, we now are in the course of performing such experiments. We haven't shown that yet for FAH and I kappa B beta, but that's ongoing now in the lab.
Okay, thank you. Any other questions from anybody in the group? If not, then Carsten, thank you. Thank you, so very interesting.
Thank you very much.
Yes, thank you. So next we have Betty Gardy, and while Betty, you are bringing up your slides, I would like to take a moment to introduce you to the community here. So Betty describes her scientific career eventful. And during this time, she has traveled a lot. But today, and now she's based in Nantes on the west coast of France, where she works in an institute that is dedicated to studying cardiovascular diseases. So she kind of found the field of hypoxia in 2003 when she first joined the lab of Stefan Richard. The lab was studying patients with von der Hippel-Lindau disease, and so this is where her interest to hypoxia then came from. And since then, she has extended her studies to patients with polycythemia. Outside work, she enjoys organizing huge parties, which is good to know, for her friends and colleagues, because what drives her enthusiasm in life and science is the collective. And aptly, her motto is, "Alone we go faster, but together we go further." So with this, Betty, I give the floor to you. Thank you.
Thank you very much. So I share the screen.
Yes, there you go. Thank you. Lovely.
Here. Yes.
Yeah.
So thank you very much for the invitation. So I will present you a European study on genetic variants identified in PHD2 and associated with erythrocytosis. So the laboratory is working on hereditary disease associated with mutation in genes of the HypoxEU pathway. So here you have the list of genes, and depending on the gene and the mutation associated, patients can develop either overproduction of red blood cells, also called erythrocytosis or polycythemia, or they can develop multiple tumors like pheochromocytoma or renal cell cancer. All the actors of that are mutated in this disease, they all are related to the activation of the hypoxia pathway factors. So today I will just focus in a study of only one gene, PHD2 gene, also called EGL1. And this study was possible thanks to two European networks, so HypoxIaNet and MPNeuroNet. And in collaboration with François Giraudon, we federate many experts in France and in Europe, experts in genetics and NGS screening, but also in diagnosis, hematology, and also expert in the hypoxia pathway. So altogether, we identified 37 new cases of erythrocytosis associated with heterozygous germline mutations in PHD2. So you have here the list of the variants, including one entire deletion of the gene. Patients present high level of hemoglobin, hematocrit, red blood cell count, but they all present a normal level of erythropoietin. We perform in silico analysis, and you have here an example of one mutation targeting the amino acid 389. And you have an example of a radar view of the different prediction tools, so like Polyfen, SIFT, etc. And the larger is the red zone, and the greater is the probability that the variant is damaging. We also perform segregation studies of the variant into families, but this study was complicated because families are usually of a small size. Women can be LC carriers due to natural regulation by menstruations, and also blood donors can be misdiagnosed. So you have here the example of a pedigree of a family carrying this mutation targeting this 389 amino acid. There is 3 carriers, and only 2 develop polycythemia. And the third one, indeed, we discovered that it was this patient was a regular blood giver, and he found it was a good treatment by chance. So we study the localization of amino acid targeted by this mutation, missense mutation. So in the lower panel, you have the published PHG2 variants, and in the higher panel, you have the new ones. And as you can see, variants can be located all along the PHG2 protein, and not necessarily in domains that can be intolerant to genetic variations indicated in red. So this in silico study was a little bit puzzling. So we performed functional studies by first using endpoint luciferase reporter assays. So we transfected cells with plasmid encoding PHG2 in addition to If2 alpha. And we could transfected the reporter luciferase assay under the regulation of hypoxia responsive element. So here you have a signal of luciferase in presence of If2 alpha. And when you add PHG2 at different concentration, you have a downregulation of this signal. And this test only show deleterious effect for only one mutant, the mutant targeting this amino acid 389 that was predicted to be highly damaging, like the others. So we choose to perform a more sensitive luciferase test by measuring the accumulation of luciferase over time. So here you have the signal in 24 hours of luciferase when only If2 alpha is transfected. The Y type totally inhibit the signal. And here you have an example of deleterious mutants already published in the literature. And all the mutants we tested, unfortunately, we didn't find any If inhibition. So you have here the in silico prediction, and we obtained the same result for them as the described polymorphism or the Y-type protein. So we said maybe we have our tests are not sensitive enough. So we asked help to Professor Kovunen and Matti Milikowski. And they perform a very sensitive enzymatic assay to measure the catalytic activity and kinetic properties of purified PHG2 variants. So they detected decarboxylation of 2-oxoglutarate, which is the co-substrate of the hydroxylation reaction. And they measured the hydroxylation of the substrate with by using If1 alpha or If2 alpha oligopeptides. Unfortunately, they didn't find any significant difference between the Y type and the mutants. And for example, for the mutants that target the amino acid 349, there was a lower affinity for these variants with a higher Km, but always compensated by a higher velocity with the Vm. So in parallel, we studied the protein stability after transfection of PHG2 expression plasmid. And 24 hours after transfection, we inhibit the protein synthesis by using a cycloheximide agent. So we didn't find, so we measured the expression of PHG2 over time with kinetics, and we didn't find any difference between the Y type and the mutants, notably for the mutants targeting this amino acid 349. But we are very happy. So first positive results for different variants. And you can see a lower expression of these variants at after 10 hours compared to the Y type. So we found loss of protein stability for the amino acid 269, the 334, at last, the one already published, arginine 371 histidine, and once more, the amino acid 389. So we decided to make an hypothesis regarding a potential impact of these variants on splicing. So here you have, so we started by studying exon splicing of exon 3. So here you have the in silico prediction regarding splicing, and for each variance tested, you have here the wild type sequence and below the mutated sequence. And for each of them, you can see that the genetic variations induce a loss of binding of splicing factors. So they all predicted an impact of splicing. So we perform a splicing reporter assay by using a mini gene. So we cloned the PHG2 exon 3 in a PCAS2 plasmid containing 2 artificial exons named exon A and B. We transfected this plasmid, we perform RNA extraction and RT-PCR by using primers located in exon A and B. And then we analyze the PCR product on agarose gel. And what we saw is here you have the correct splicing. So exon 3 spliced with exon A and B. And here you have a splice with exon A and B only. And we didn't find any difference between the wild type and the mutants. So we performed the same experiment with the exon 4. So we studied a mutant just located at the beginning of exon 4, which remove a site of splicing factor binding. And this mutation is called, I mean, there is no impact on amino acid change. So we call this mutant a synonymous mutant. And the second mutant is located at the end of the exon 4 in the splicing donor site. The G is replaced by a T. We perform a splicing reporter assays, and this time we observe a strong effect of this variant, the variant targeting the splicing donor site, with a total exclusion of exon 4, so total exon 4 skipping, and only a splicing of exon A and B. And regarding the synonymous mutant, we find a very slight impact with a band always of higher intensity, but not very dramatic. So we study this exon 4 splicing on patient cells. So we extract RNA from gallblood by using a PacGene tube. And we also study some lymphoblastoid cell lines. So we confirm this strong effect of the variant targeting the splice donor site with this exon 4 skipping here. But still, we only have a very faint effect of this synonymous mutant here and here. So we we purified, clone and sequenced this band. We confirm that this band corresponds to the exon 4 skipping. And so the consequence is that when the exon 3 is spliced with exon 5, you introduce, the exon 5 introduce in phase premature termination codon, a stop codon, that lead to a truncated protein at amino acid 382. And this protein is theoretically non-functional. So we really focused on this patient with this synonymous mutation, and we decided to use iPS cells, so induced pluripotent stem cells, reprogrammed from the patient. And we differentiated this iPS into hepatocyte-like cells. After 22 days of differentiation, we put the cells 24 hours in hypoxia, and we perform RT-PCR by using primers located in exon 3 and 5 to measure the exon 4 skipping. And we quantified this the different isoform by migrating the PCR product on the TAP station. And here you have the quantification in normoxia and hypoxia. So the white type in blue and the mutant, Cyanogenix mutant in orange. And we demonstrated that the exon 4 skipping is reproducible, and we can see high level of this transcript in the patient cells in normoxia, but also in hypoxia. And the very interesting point here is that we discovered that this PHD2 exon 4 skipping, in fact, is increased in hypoxic normal condition here in blue. So we analyze RNA sequencing data already available in the lab of these white type cells. So here you have a zoom of the exon 5, 4, and 3. And you can see that this RNA sequencing data confirm this exon 4 skipping with a lower expression of exon 4 in hypoxia. So in conclusion, this study of these 37 PHD2 variants show that unfortunately in silico tools are not always reliable. The functional studies allowed the classification of 10 variants as deleterious, so the entire deletion, 3 variants with truncated proteins, 1 variant with a loss of function and protein stability, 4 variants with a loss of protein stability, 1 variant which impact splicing, and the last one, this effect is very faint, so we call this variant hypomorphic variant, which has an impact on splicing with a consequent truncated proteins. So in the future, we really need to study a precise mechanism of PHD2 splicing, and notably to study the role of this exon 4 skipping. And this questions the role of the PHD2 in hypoxia, because we all know that PHD2 is the If target gene. So if more than 60% of this PHD2 mRNA is not spliced correctly and the protein is not functional, so we may think about this. But we also need to study PHD2 expression. So for example, we have a variant located in the promoter in -410, just targeting the If binding site. But also, we need to study other partners of PHD2. And for example, we have 2 variants, arginine 35 and asparagine 50, located in the zinc finger domain, known to bind the cochaperone P23 and linked to the If stability. So once again, thank you very much to all my collaborators, and I'm ready for your questions.
Thank you, Betty, for a lovely talk. So we have a question here from Alex Suvak, who's asking, since women are healthy carriers due to regulation by menstruation, do you think that the risk of erythrocytosis could increase after menopause?
Yes, of course. And we in fact, when we perform family history questionnaire, this woman remembers that when they were young, they may have erythrocytosis, which stop at puberty. And this woman came back to the doctor after menopause because the erythrocytosis is back again.
Can I ask a question?
Yes, please.
I'm very sorry for not using the chat. But
no, that's better. Please go ahead.
Betty, very interesting. Obviously, the obvious question is this a hypoxic effect something to do with PHD2 catalytic activity, or does the signal have another source? So I wonder if you've had the opportunity to test whether a relatively specific PHD2 inhibitor like roxadustat had this effect, and whether that differed from a non-specific 2OG analog such as DMOG?
No, we didn't test this, and we plan to. Yes, because we...
That could be very simply done, and of course, it'd be very interesting if it was something other than PHD2.
Yes.
Are there any more questions? If anyone would just like to shout out. Sorry, it's difficult to see. I was wondering about PPGL formation and whether you observe this with any of your variants.
Yes, so we only describe one case with paraganglioma, and this case, these patients carry a very severe mutation. So we still don't know if he developed paraganglioma because of this variant, which is very deleterious. Is one of the 3 coordinators of iron binding, or if these patients carry another susceptibility gene of paraganglioma development. But this is the only one case described. So now there is more, almost 100 of patients described with PHD2 mutation, and we didn't observe any more case.
Thank you. So if we don't have any more questions, then perhaps we should move on to our next speaker.
Yes, thank you. Thank you, Betty, and thank you, Tammy. Yes, we're moving on. Next speaker, we have Deepika Watts. And Deepika, while you're preparing your slides, I will give a little bit of background. So Deepika grew up in India, but she moved to Sweden for her master's in molecular biology. And after she obtained her master's, she continued working at the Karolinska Institute before moving to Dresden in Germany for her PhD studies. She's focused on studying the role of T regulatory cells in type 1 diabetes. And in fact, before she kind of joined the field of hypoxia, she majored in immunology. Today, she's a postdoctoral fellow in the lab of Professor Ben Vilox at the medical faculty of the Technical University of Dresden. So now Deepika enjoys working on different research topics. She believes that the complexity of biological and physiological systems lies in the interplay of all these systems involved, which is possibly quite true. Outside work, she loves to travel because, and I totally agree with you, Deepika, here, she feels, as I do, that traveling is one of the best ways to have real life experiences. And also because, you know, that's there you learn a lot more that you don't actually learn in the lab. So agree with you on that one, Deepika, and please, the floor is yours. Thank you.
Thank you, Krista, for the introduction. I'm trying to switch to the...
Yeah. We can see you on presenter mode now. There we go.
Yeah, I was just trying to switch to the laser pointer, but it's not working. That's fine.
Okay.
So thank you, Krista, for the introduction. And thank you to the organizers for this very nice initiative of bringing the hypoxia community together, especially in these Corona times. I think this is a very nice platform for us to share our work. And so today I will be presenting my work on the role of PHD2 and HIF1 axis in the adrenal gland steroidogenesis in the mice. As Krista mentioned, I come from the group of Professor Ben Vilox, the unit of hypoxia signaling in vivo. We are a group that's majorly focused on studying the hypoxia pathway proteins. On top of our research are our mice, because we are a highly in vivo group. We have several floxed lines with hypoxia pathway proteins. We are interested in studying the different physiological aspects And with specific retargeting of cell types or the tissues, we have our focus on different aspects such as hematopoiesis, erythropoiesis, adrenal gland, bone metabolism, and chemokinesis. In addition to that, we also have several tumor and metastasis models established in the lab and skin wound healing model that we use for our research. The group has successfully established the significance of hypoxia pathway proteins in different fields in the past years with significant amount of publications on the different topics and how important the hypoxia pathway proteins are for the maintenance of physiological and pathological conditions. I have majorly focused on the adrenal gland biology in the past years. So in the next few minutes, I would just like to convince you how important the adrenal gland is and how the hypoxia pathway proteins are important for the steroidogenesis regulation in the adrenal gland. So we, on the daily basis, encounter stress. We all use this term more often than we would like to use it. But how does our body react to stress, and what is the major role of adrenal gland in response to stress? So when our brain senses any stress signal, the signals perceived by the brain send the signal from hypothalamus to the pituitary gland, and pituitary releases this adrenocorticotropic hormone in the bloodstream, sending the signal to the outer layer of the adrenal gland, the adrenal cortex, which in turn leads to the release of steroids. The corticosterone and the aldosterone, the two major hormones released from the mouse adrenal cortex. On the other hand, then brain senses the signal, the sympathetic nervous system also sends the signal to the inner part of the adrenal gland, and that releases the catecholamines. Those are also essential for the stress management. All these hormones are essential for the physiological conditions, for the regulation of heart rate, blood pressure, and inflammatory immune response, and many more aspects which I have not listed here. So, but how do the PSDs and HIFs, the hypoxia pathway proteins, impact the release of catecholamines and the steroids in the adrenal gland has been the interest that I have been working on in the past years. And we have successfully established that the hypoxia pathway proteins are very important for both the catecholamine release and the steroid release. However, today I will be talking about the role of hypoxia pathway proteins in the release of corticosterone and the aldosterone. So when I started my research, there was not much known about the research in the hypoxia pathway protein research in the adrenal gland in vivo. There were several in vitro studies from the cell lines with treatment with hypoxia, or some studies where rats were kept in hypoxia and the effect on the adrenal gland was studied, or some in vivo studies in the zebrafish. However, there was nothing with the manipulation of hypoxia pathway proteins in the mice. So for the first step, we generated the AKR1B7 tree-specific, cortex-specific mouse line with the ablation of PSG2 and HIF1 specifically in the adrenal cortex. So these mice will be referred to as P2HIF1 in my rest of the presentation, and these mice gave us the chance of studying the impact of HIF1 downregulation and HIF2 upregulation on steroidogenesis. All the results from these mice will now be depicted in red. So first, to see if the pre-targeting of the mice, we crossed them to the reporter strain, and we found that all the cortex was specifically targeted in these mice strain. So first, we wanted to see if there is any effect on the morphological changes, or there was any structural impact on the adrenal cortex. For that, we performed the paraffin adrenal stainings for the H&D stainings, and we found that there were no differences, any obvious morphological differences on the paraffin sections. Then we moved on to the cryo sections, and interestingly, to our surprise, we found that the lipid content in the P2HIF1 mice was significantly reduced. So why is the lipid content important? Because the lipid is the first step of the this is the precursor for pregnenolone, and the conversion from cholesterol to pregnenolone is the first step on the start of steroidogenesis. We wanted to now see if this change in the lipid content is also related to the amount of steroids produced in the adrenal gland. So with mass spectrometry, we measured the steroids in the adrenal gland, and we found a significant increase in all the steroids, and the precursors of the steroids increased in the adrenal gland. Interestingly, the two major the corticosterone and the aldosterone, two major hormones released by adrenal cortex, was significantly increased in the gland. We also checked the plasma of these mice, and we found a significant increase in the three hormones in the plasma as well. So since we saw this increase in the steroids, we thought there could be also an increase of the transcription analysis. We wanted to see the enzymes and the proteins involved in the pathway, if there is also an increase in these enzymes. For that, we isolated the mRNA from the whole adrenals and looked at the enzymes expressions. So STAR is the rate limiting step for the conversion of progesterone, and CYP11A1 are the two important enzymes. Those were significantly increased. However, also the other enzymes involved in the pathway were increased. Next, we wanted to, because we had the reporter strain, we had the chance of isolating the GFP positive cells from the wild type mice and the P2H1 knockout mice. So by implementing the flow cytometry sorting, we isolated the GFP positive mice, GFP positive cells from these two, the controls and the knockout mice, and performed the bulk RNA sequencing. And interestingly, we found several of the gene signatures related to the increased steroidogenesis. For example, the cholesterol biosynthesis, the glucocorticoid metabolic processes were significantly increased. Also, the cholesterol homeostasis and the response to the mineralocorticoid, the aldosterone, was increased in the cells specifically lacking the PSG2-HIF1. Next to that, we also found several gene signatures. Those were positively regulated in the HIF1 deletion fashion, and that was also something very interesting. Next to that, we also found the beta-catenin pathway that was upregulated and is responsible for the hyperplasia in the for the hyperplasia of the adrenal gland, adrenal cortex especially. So we could establish that the P2H1 mice with HIF1 downregulation and HIF2 upregulation were important for the increase of steroidogenesis. So but next, we did not know if the impact that we were seeing on steroidogenesis was HIF1 mediated or HIF2 mediated. So we wanted to see if the effect observed in these mice is coming because of the HIF1 downregulation or the HIF2 upregulation. For further investigating this, we created another mouse line, the AKI1B7 pre-PSG2, PSG3, which gave us the chance of studying the impact of HIF1 upregulation and HIF2 upregulation. So HIF1 is an inverse factor in this case. These mice will be referred as PSG P2 and P3 in the rest of the presentation, and the results from these mice are depicted as yellow. So we wanted to now see if these mice also have any impact on the steroidogenesis. And interestingly, we found that the three major steroids released from the adrenal cortex were significantly reduced in the adrenal gland in these mice, and also the plasma depicted the same results with the decrease of progesterone, corticosterone, and the aldosterone. So we assumed that HIF1 alpha is regulating the steroidogenesis inversely. Also, in the same line as we did for the P2H1 mice, we checked for the transcription analysis here for the enzymes involved in the pathway, and several of the enzymes, genes involved in the steroidogenesis were significantly reduced, and it went in hand with the overall reduced steroidogenesis. So the HIF1 upregulation led to a decrease of the overall steroidogenesis pathway, the enzyme, and the production of the steroids. Next, so just to conclude here once, so HIF1 downregulation and HIF1 upregulation led to HIF inversely regulated the steroidogenesis, the genes involved in the pathway, and the steroid release from these mice was inversely regulated by HIF1. So we believe that HIF1 is the major regulator of the steroidogenesis in the adrenal gland of the mice and inversely regulates it. Next, we wanted to see if, since it's known that glucocorticoids have very anti-inflammatory effect and are commonly used as immune suppressants and infect the immune system in a negative way, we wanted to see if there is also the case in these mice with an increase of steroids or decrease of steroids. So first we checked the P2F1 mice with the mesoscale discovery. We looked at the different pro-inflammatory cytokines, and there was a significant decrease in a lot of cytokines, which was very interesting because it depicted the change of the glucocorticoids was actually impacting the immune system. However, there was the chemokine CXCL1, which was significantly increased. It is established that this is a chemoattractant for neutrophils and helps in the recruitment of neutrophils in the bone marrow. Thus, and also the glucocorticoid increase or the treatment with glucocorticoids leads to the effect on neutrophils. So there was an increase in the circulation of neutrophils in these mice that went in hand with the increase of glucocorticoids. On the other hand, glucocorticoids also affect the lymphocytes and the eosinophils, as is known in the literature, and we saw the similar decrease of lymphocytes and eosinophils here. We also decided to check in the P2, P3 mice if we observed the similar changes, and we observed that there was an increase in some of the cytokines. Also, those are involved in the pro-inflammatory process. But just it was not that the end that we found that the change in this glucocorticoids because of the HIF1 mediated changes led to the changes in the immune system. We also observed next to the changes in the immune system that there was an effect on the hematopoiesis, the hematocrit and the hematohemoglobin of these mice. So this was not the part of the study, but it was something that came up because we were just interested in looking up what are the effects on these mice. And while keeping the mice for the longer periods, we observed there was some P2H1 mice, those were pale as compared to the wild type controls, and we measured the hematocrit hemoglobin levels in these mice. Interestingly, the hematocrit and the hemoglobin levels in the P2H1 mice were significantly decreased, and in the P2, P3 mice, they were significantly increased. So from this point, we concluded one thing that, of course, HIF1 is a very important factor for the steroidogenesis, and it inversely regulates the steroids production in the adrenal gland, but that's not it. So there are further impacts of the HIF1 downregulation or upregulation in the adrenal gland that can lead to the effect on immune system and hematopoiesis or erythropoiesis. So at the moment, we are very interested and we are looking at the impact of all these changes. Those come from the hypoxia pathway protein changes in the adrenal gland on the hematopoiesis or the erythropoiesis, and also the immune system. So we are also interested in performing some experimental disease models in these mice and see how these mice react to the diseases, to the any disease models. So this is the current, this is the conclusion of the project and the current focus of the research. With this, I would actually like to thank Professor Ben Wheelox for his continuous support and motivation and the guidance for the project. All the group members of past and present who have worked or supported in this project, all the collaborators, and I would like to thank you all for attention, and I would be happy to take any questions now.
Thank you, Deepika, for a very interesting talk. We have a question here from Sylvia Martin-Puig, who is asking whether you have looked to evaluate the direct role of HIF2 in steroid induction by treating your PHD2 HIF1 knockout mice with the HIF2 alpha specific antagonists.
No, we have not done that. We have not treated these mice with the HIF2 specific inhibitors. We have only, because we did not see any effect that was HIF2 mediated, but that's an interesting point, but we haven't done that.
On a related note, I suppose it's interesting, do you have some theories about how HIF1 might be negatively regulating your steroid-related enzymes, if you think it's not HIF2.
So yes, we actually have a theory for that. We hypothesis for that, and we are also interested in investigating this further. We believe that this is, of course, because this was the first question that came to our minds. Because HIFs are usually the induction factors, and you always expect a positive regulation from them. But we believe there are some MIRs involved. There are also some MIRs known. Those are known for regulating the steroids. Like they are responsible for the regulation of steroids. We believe these MIRs are the responsible elements for this inverse regulation. So these are the ones, those are being upregulated by HIFs and leading to the downregulation of the steroid production. And we are currently interested in investigating that.
Have you tried temporal inactivation of PHD2-HIF1 to see whether these are developmentally induced defects or?
No, we have for this project. No, we have not done that.
And did you see any effects on blood pressure in these mice or on PNMT levels?
We did not measure the blood pressure levels. We checked the PNMT levels, but the PNMT levels were not changed in these mice.
I don't know if anyone else has any questions. Just shout out.
If not, we can move forward. And again, please feel free to also, I'm sure the speakers are happy to answer questions afterwards as well, you know, if something comes to your mind, so feel free. Okay, our fourth speaker today is Blanca Jimenez. And again, Blanca, thank you for being here today. And while you're preparing your slides, I will give a brief introduction. So Blanca was born in Granada, which is a beautiful city in a southern part of Spain. Been there, beautiful, absolutely. She majored in biology at University of Granada before she moved to Madrid to do her master's in genetics and cellular biology. Currently, she's studying for her PhD at the University of Seville in the group of Jose Lopez Baneo. And the main research area of the group is the study of oxygen regulation of breathing. And in particular, the group is interested in the reflex ventilatory response induced by hypoxia. Outside work, Blanca loves cooking all sorts of dishes. You know, that kind of reminds experiments in the lab, doesn't it? And she's also an outdoorsy person whenever possible, and also loves traveling. So Blanca, happy to have you here today, and please
thank you.
Take the floor. Thank you.
Could you see that?
Yes, we can. Thank you. Yes.
Okay.
There you go. And present. There you go. That's good. Thank you.
Okay. Thank you, everybody. It's a pleasure for me to share with you the result of our research. And as you can see from my title, I'm going to talk about how the oxygen regulation of breathing is restored by NDI1 expression in mitochondrial complex 1 deficient mice. Wait for that. So acute response to hypoxia as hyperventilation and sympathetic activation are mediated by the carotid body, the most important chemoreceptor. The carotid body is located in the carotid artery, and his function is essential in acute response to hypoxia. The carotid body contains a specific neurosecretory sensory cells called as glomus cells, which contain a specific oxygen-regulated ion channels, which inhibition during hypoxia produce cells despolarization, calcium influx, and release of transmitter, which activate different sensory fiber, conveying the information to the respiratory center, increasing the ventilatory in response to acute hypoxia. But the mechanism underlying this oxygen sensing of these carotid body glomus cells had been elusive, but recent experimental data suggests that this glomus cell contains an highly specialized and activated mitochondria able to sense hypoxia. In 2003, in our group, we monitored the neurosecretory activity of these carotid body glomus cells by the amperometry techniques. And as I mentioned before, during hypoxia, there is an increase in the secretory activity of these carotid body glomus cells. We use different electron transport chains blocker. In contrast, we observe that the rotenone, a block for the mitochondrial complex 1, mimics the effects as hypoxia and also occludes the response induced by hypoxia. So the rotenone acts in the ubiquinone binding site in the mitochondrial complex 1, blocking the NADH kinase oxidoreductase activity. So this data suggests us that this mitochondrial complex 1, in particular, this ubiquinone binding site could have a key role in response to acute hypoxia. So for that reason, we decided to generate a knockout animal by the elimination of one of the 3 subunits that form this ubiquinone binding site. For that, we use a Krylov-XP approach in order to eliminate NDUFS2 using a specific recombinase under the tyrosyl hydroxylase promoter by elimination of this NDUFS2 in Th-positive cells specific for catecholaminergic tissues. So we first studied the hypoxia response in whole body animals by plethysmography, and you can observe here when there is a decrease in the oxygen tension, there is a rapid increase in the ventilatory frequency in the control mice. But in contrast, in the cow animals, the response is practically abolished. So this data suggests that this mitochondrial complex 1 have an important role in acute response to hypoxia, systemic levels. So next, we decided to study also the neurosecretory response in these carotid body glomus cells. And you can observe here that during hypoxia, as I mentioned before, there is an increase in the neurosecretory activity of these carotid body glomus cells. And we use another stimuli to confirm that our cells were alive and were normally. And in contrast, in carotid body glomus cells from these knockout animals, we can observe that these effects are completely loose. These glomus cells were insensitive to hypoxia, despite the fact they are sensible to another stimuli as hypercapnia. So this data suggests that the mitochondrial complex 1 is important in acute response and also in the neurosecretory response to hypoxia in the carotid body glomus cells. So next, we decided to study which molecule could modulate the ion channels, and we proposed to candidate, we proposed the NADH as a substrate of the mitochondrial complex 1 and also the reactive oxygen species which are produced by the mitochondrial complex 1 and for the mitochondrial complex 3. So we monitor the production of the NADH and ROS as a signaling molecule, and by microfluorimetry techniques, we could record the increase in this NADH and the ROS in the intermembrane space during hypoxia in carotid body glomus cells. And as we have expected in carotid body glomus cell knockout for this NDUFS2 for the mitochondrial complex 1, we can see that this effect disappears. So all data suggest and give us the opportunity to propose this mitochondrial to membrane signaling model of acute autosensing. We propose that hypoxia produces a backlog of electrons into the electron transport chain, and it causes the accumulation of reduced ubiquinone, and that leads a slowdown or even a reversal activity of the mitochondrial complex 1 by producing this NADH and ROS signaling molecule that could modulate these ion channels. But recent experimental data from another groups propose also that the structural shift of the mitochondrial complex 1 to a deactivated state of the reduction of nathistane in the UFS2 subunit, they suggest that it occur during acute hypoxia. So we wanted to know the role of this mitochondrial complex 1 in this carotid body glomus cell, and the question that we planned was is the mitochondrial complex 1 have oxygen sensitivity, or it is simple, has a signaling role in response to hypoxia kinome reduced accumulation. So there are numerous genetic tools that could allow us to answer that question. And one of them was the expression of NDI1. NDI1 is the main entry point into the electron transport chains in yeast, and as the mitochondrial complex 1 catalyzes the oxidation in the matrix of the NADH, but it does not pump protons to contribute to the production of ATP. So in absence of the mitochondrial complex 1, we propose that NDI1 could oxidize NADH and maintaining a functionality electron transport chains. So we propose that the replacement of this mitochondrial complex 1 could produce the same effect during hypoxia as the mitochondrial complex 1. We propose that during hypoxia, as I mentioned before, it happened a backlog of electrons into the electron transport chain, and the NDI1 could produce this NADH and ROS signaling molecule and could restore the sensitivity to hypoxia. So for that reason, we decided to generate a knockout animal for NDUFS2, but with the expression of NDI1 as a knocking mice, in order to understand the function of this mitochondrial complex 1 in response to acute hypoxia. So the expression of NDI1 and the elimination of NDUFS2, we create this animal using a QuiLOX-P approach, also using a specific recombinase under the tyrosine hydroxylase promoter for removing NDUFS2 in catecholaminergic tissues and with the expression of NDI1. The expression of NDI1 is associated also with the expression of a green fluorescence protein as a marker. So we first decided to study if our model was working correctly. So we first performed an immunostaining for this green fluorescent protein to confirm that we were expressing NDI1 in these carotid body glomus cells, and you can observe in the cow NDI1 animals there is an expression of there is a marking of green fluorescent protein in Th positive cells. So we were expressing NDI1 in catecholaminergic tissues as carotid body. So next we also want to test that the expression of NDI1 did not alter the elimination of NDUFS2. So we performed an experiment using different samples of carotid body from different group of animals, and we first studied the mRNA level of NDUFS2. We can confirm that the expression of NDI1 did not alter the elimination of NDUFS2 at mRNA level. And also we performed an immunostaining to confirm that propose at protein levels, and you can observe here in the wild type mice they express NDUFS2 in Th positive cells, and as I mentioned before, in the cow animals we remove the NDUFS2, but when we express NDI1 we also eliminate NDUFS2. So NDI1 did not alter the elimination of NDUFS2 in Th positive cells. So next we have confirmed that our model worked correctly. We first studied the whole body response to hypoxia by plethysmography. And you can observe in the wild type animal, when there is a decrease in the oxygen level, there is an increase in the ventilatory response in the wild type mice. As I mentioned before, in the cow mice, in mitochondrial complex 1 deficient mice, the response was completely loose. But in these cow animals, when we express NDI1, we can observe practically a completely restoration of the ventilatory response to hypoxia, as you can observe in the quantification below. And here in the panel of the right, we can observe a typical response to hypercapnia in all three groups. So this data suggests that the NDI1 is enough to restore the sensitivity to hypoxia, the ventilatory response in these mitochondrial complex 1 deficient mice. Also, we wanted to know whether NDI1 could restore another systemic parameters. So we first studied the red blood cell proliferation as a chronic response to hypoxia. Here in blue, in the wild type mice, when they were under 10 days under hypoxic condition, there is an increase in the hematocrit, but in contrast, in the cow animals, Due to the lack of hypoxyventilatory response, the increase in the hematocrit is significantly higher to the wild type. But in these cow mice, when we express NDA1, we can observe that we restore, as I mentioned before, the hypoxyventilatory response, and also we restore the increase in the hematocrit similar to the wild type mice. Also, we have observed that in the cow animals, there is an increase in the lactate in normoxic condition in blood, and we propose that it could probably happen due to the lack of the mitochondrial complex 1. This animal could have accelerated glycolysis. So in this mitochondrial complex 1 deficient mice, when we express NDA1, we can observe that there is a complete restoration of the basal of lactate in blood in normoxic condition. And to finish with all systemic data, I want to show you this amazing picture. And we can observe in the cow animals, they show dwarfism and they die before 2.5 months. We propose that it could probably happen due to damage in Th-positive hypothalamic and another important central neurons. And in contrast, in the cow NDA1 mice, we can observe that there is a complete restoration of the size of the animals, and also they live normally until they were used for experiment. So this ability of NDA1 to rescue other systemic phenotype open the possibility to use NDA1 to restore dysfunction of mitochondrial complex 1. And next, I want to show you that we have studied the neurosecretory response to hypoxia in this animal as well. And as I mentioned before, during hypoxia, there is an increase in the secretory activity of carotid body glomus cells, and this response was completely lost in these cow animals. But in these mitochondrial complex 1 deficient glomus cells, when we express NDA1, we can observe that there is a restoration of the hypoxia neurosecretory response in these carotid body glomus cells. So NDA1 could restore systemic ventilatory response and also the neurosecretory response in mitochondrial complex 1 cow animals and cow carotid body glomus cells. So to finish, we also measure the chemosensory response of these cells by microfluorimetry techniques. And as I mentioned, we propose two important signaling molecules, NADH and ROS, which modulate the ion channels. And as I mentioned before, in the wild type animals, during hypoxia, there is an increase in the production of NADH, and this response practically disappears in the cow animals. But when we express NDA1 in this cow animal, in this cow carotid body glomus cells, there is a rapid and reversible, faster and higher production of NADH during hypoxia in these carotid body glomus cells. So NDA1 restore, even enhancer, the production of NADH response to hypoxia. And we also confirm the production of ROS in the intermembrane space. And during hypoxia, there is an increase in the production of ROS in carotid body glomus cells from control mice. And this response was lost in cow cells. But in these cow cells, when we are expressing NDA1, we can observe a complete restoration of the production of ROS during hypoxia. So to conclude with all this data, we propose that NDA1 in our mitochondrial to membrane signaling model acts as an effector because the hypoxia produce a backlog of electrons, and it causes the accumulation of reduced ubiquinone, and it leads and slow down or even a reversal activity of the NDA1, and NDA1 produce these two signaling molecule, NADH and ROS, which modulates the ion channel. So in our model, we can confirm that the mitochondrial complex 1 acts as an effector, and it could be replaced by NDA1, and we could restore the sensitivity to hypoxia. So next, I want to show you our principal conclusions. The first one, the transgenic NDA1 expression completely rescued the systemic hypoxyventilatory response in mitochondrial complex 1 deficient mice. The cellular and mitochondrial responses to hypoxia are fully restored by NDA1 expression in mitochondrial complex 1 deficient carotid body glomus cells. So this data strongly suggests that the mitochondrial complex 1 signaling is essential for acute responsiveness to hypoxia in arterial chemoreceptor cells, independently of this bioenergetic function. Mitochondrial complex 1 does not have intrinsic oxygen sensitivity. And NDA1 expression prevents early death, dwarfism, and other general alteration in the mitochondrial complex 1 deficient mice. And as a future perspective that we propose is that transgenic NDA1 gene expression could be a potential therapeutic approach to restore mitochondrial complex 1 dysfunction in human pathology, such as Leigh syndrome or Parkinson's disease. And to finish, I want to thank you to all my lab, especially to Jose Lopez Barneo, and thank you everybody for your attention.
Thank you, Blanca, for that very interesting talk. We have a question here from Daniel Dietrich, who asks, did you see an accumulation of succinate? See the science paper, 3rd of December, 2021.
I don't know how do you exactly want to know, but if we have seen an accumulation of succinate, but in what model, in the cow animals or in the cow and the A1 animals?
I don't know, Daniel, if you want to come on the call and explain your question.
That's fine. Hi. Well done, first place. So that's actually very excellent. In the paper just that I cited, they actually showed that under low oxygen conditions, succinate will accumulate because the electron transfer chain going backwards toward the MC2 actually accumulates succinate because it takes up the excessive electrons. So the question is, what is actually going on in your knockout mice with the NDA and NDI1?
We have not checked the level of succinate, but we have performed also experiments using dimethyl succinate to increase the kinome reduce, and we have observed that there is a potential of the hypoxia activity, the neurocirculatory response to hypoxia. So we think that the accumulation of reduced ubiquinone or by the hypoxia or introducing succinate, it could increase the secretory response to our cells, but we have not checked the level of succinate in this model.
Okay, thank you very much. Thank you. Gracias.
Thank you.
Thank you. Are there more questions coming through? Please just make yourself known. Amuli, is that the question? No. I was wondering whether you think these principles of NDI1 and mitochondrial complex 1 apply to other acutely oxygen sensitive tissues like the pulmonary arterial smooth muscle cells.
I'm not sure what do you mean? I don't understand well.
I was just thinking about whether you think NDI1 would rescue the phenotype of MC1 and deficiency in other cell types that are acutely oxygen sensitive.
I think that NDA1 could also restore another groups have studied how NDA1 could restore the ataxia in another mouse models. So I think it's a good potential approach to rescue these mitochondrial complex 1 deficient pathologies.
Thank you. Yeah, I was thinking about expression of alternative oxidase in pulmonary oxygen sensing.
Anyone else? We now have time for extra questions. Now, all the speakers were excellently within their time limit, so thank you. So if anybody has any other questions, just raise your hand and feel free. If not, I also thank you, Blanca, and all the other speakers. Here we have time to, open the floor for questions and discussions. If I know it's quite tricky to, when we are here virtually, but if you have something that you want to bring up to the team or ask questions or anything else. So now is a good moment. We do have time. So quite quiet over there. Okay, no worries. Totally get it. While I have you here, I of course I have few notes and things to inform you. So we're going to have the next HypoxEU in March. So if you or somebody from your group or somebody you know wants to present their work, just please contact me or anybody in the scientific organization committee. So we'll be happy to get new speakers and ideas and of course ideas to how to further develop this program, this community. So that's one. Another note that I have here for myself is that we're planning on having a live HypoxEU in the latter parts of 2022. So that's something to look forward to. And this is why we're building up this community, because one day, believe you me, all this will be over, this whole pandemic, and we can again get together. And when that happens, then we'll all already know one another and it'll be all the more fun. I want to thank the speakers, excellent work, excellent research, excellent responses. You managed well. Our more experienced members of the audience, you had tough questions, but the speakers managed really well. I want to thank the Scientific Organization Committee. If anybody of them wants to say something, Roland Wenger, Randall Johnson, Dr. Kaczynski there, Chris Pugh, Cormac Taylor, Edurne Berra, Johanna Myllyharju, Peppi Karppinen. So thank you again for supporting the action here. You're wonderful. And if nobody has anything to say here, I kind of hope that I would propose that everybody goes off mute, just unmute yourselves and would just give a big hand, but I don't know if that's going to be a huge disaster. But should we try what happens if we all give a big hand? I don't know how that works, but if nobody has anything to say, I do have one thing to say. I want to wish you all a very Merry Christmas time here from Finland, and I'll see you next year.